Good afternoon, everybody. Thank you so much all for being here. Today, I'm going to be talking about some current work of mine during my PhD time at Brown University. The title of my project is called WINDEX, a hierarchical integration of site and window-based statistics to model the footprint of positive selection. To get us all oriented with this topic, I'd like to start by talking a little bit about adaptive mutations. A mutation can be considered adaptive when it confers some kind of fitness benefit in a population. What does this look like? Let's say we have a generic population of individuals, and let's say there's one individual that has some kind of beneficial allele that might confer to longer chance of survival or better chance of reproduction or something of other beneficial in the population. This individual or this allele in the population would face evolutionary pressure over the time. We'd expect to see a rise in allele frequency of the selected allele. This phenomenon is called positive selection. So after this phenomenon, over some time, we'd expect to see a rise in allele frequency of the selected allele. And a lot of times this can also be coined as positive selective sweeps. What does this look like at the genetic level? Positive selective sweeps are detected by various signals in population haplotype data. So in the pre-sweep conditions in the same population I was talking about on the previous slide, we'd expect that there would be a lot of variation within individuals in a population, and a lot of this variation would be evolving neutrally, meaning that there wouldn't be any evolutionary pressure on those individuals. However, I did mention that there's one individual that contains a selected variant, denoted here in the red box. And so we would expect in a positive selective sweep, post-sweep condition, that these conditions in a population in later generations, this would change. First, we'd see a rise in frequency of the selected allele, meaning that more individuals would carry this allele because it's beneficial. And additionally, linked alleles close by would also rise in similar frequencies, while diversity near the selected allele will decrease. So instead of thinking about the selected allele being inherited by itself, we can think about the allele being inherited in haplotype blocks. And so this poses a challenge for scientists like us that are trying to determine which alleles are selected and potentially adaptive, if it's hard to determine if signals are linked or selected in a population. So this brings us to a longstanding question in PopGen, which is, how can we model the footprint of positive selection by learning linked, neutral, and sweep signatures? And there's been mountains of work that has been done in this field over the last 40 years, starting with wonderful statistics that many of us in this room are probably familiar with. These statistics look at deviations from neutrality in haplotype data, allele frequency comparisons, and haplotype homozygosity. And this graphic is very much a generalization of what the stat might look like, but to orient people that might not be familiar with some of these statistics in the room, we would expect that these statistics would rise in magnitude and somewhat of like a bell-shaped curve as you get closer to your selected allele position in your haplotype data. I'd like to draw your attention to this emission scale column. We notice that some of these statistics are better off to be calculated in a window or a site level. And these emission scales pose a little bit more of a challenge for the field because to this day, there are no existing tools that site and window-based statistics for this multi-class classification problem, AKA learning neutral, linked, and sweep signatures. And so with that, I'd like to introduce my tool called WINDEX. WINDEX is a hierarchical hidden Markov model, or an HHMM, that incorporates multiple statistical resolutions to classify positive selective sweeps. This is a high-level overview of the state diagram of WINDEX, and I'd like to run through a couple of key points that I think you guys should take away from this. First, WINDEX takes in both site and window-based statistics, measuring positive selective sweep signals. Some of the stats that I mentioned on the previous slide could be used as input, but you input both at the window and site level according to your haplotype data. Next, WINDEX hierarchically classifies haplotype data in a sequential order from the start to the end of a genetic sequence or a genetic region. So what's important to take from here is that WINDEX operates at two main levels, level one being the window level, meaning that it learns from the distributions of the window statistics to classify the data at that window resolution. And then at the site level, these individual site state diagrams are deterministic. So for example, if you're in a window that's classified as neutral, the sites within that window can only be classified as neutral site states. And then finally, WINDEX emits both site and window-based classifications representative of all input statistics using the likelihood framework. And this likelihood framework, I don't have a ton of time to go into detail today, but if you're interested, it's adapted from Sugden et al. So I know it's probably a lot to soak in, so I'd like to walk through a very generic example of how WINDEX works. So let's say we have a 25 base sequence of variants that we'd like to classify as neutral, linked, or sweep. And WINDEX doesn't know this label, but in the room, we know that this center position here is going to be the location of our selected allele, and we want to see how well WINDEX does at classifying. The first thing that we would do is that we would bin our data into non-overlapping windows and then use those windows to calculate our choice of window statistics. And one thing that's nice about WINDEX is that you can choose the window size depending on which window statistics you're going to use and also the size of your haplotype data. So next, we'll calculate both our site-based statistics and our window-based statistics, again, in those non-overlapping windows, and then that file will be used as the input to WINDEX. And then if I were to demonstrate what an optimal hierarchical state path would look like for WINDEX, we'd start by initializing our model. Next, let's say for window 1, we move into a neutral window state, and then the sites within window 1 move into neutral site states. The probabilities propagated from both the site and the window level are then used to make the next classification, or the next transition, I should say, at the window level into a linked window state. And although there's multiple paths that the site states can take, let's just say in this example that the linked window state transitions the site states only into linked. And then as we get closer to the location of the selected allele in our data, we would expect WINDEX to transition into a sweep window state. And then at the site level, we can see transition between linked and sweep states, therefore localizing the position of the selected allele in our data. Then following the rest of the expected pattern of a positive selected sweep, we'd expect linked data to be on the other side of our sweep window and sites, and then transition back into neutral data when we're positionally far away from the selected allele location. As HMMs typically work, we would expect that each of these states would emit some kind of observation for the variance. And so let's say based on the emissions, we can observe that the windows are then classified into these categories: neutral, linked, sweep, linked, and neutral. And then based off the state information, we can say that the states are then classified into what is now filled in into our 25 base sequence of variants. So not only is WINDEX intended to be good at localizing the position of where the selected alleles are data, but also to get an estimate of the breadth and the strength of what the linked region looks like in our haplotype data as well. So next, I want to talk more about some empirical data that I generated to evaluate WINDEX. And before I do that, I want to briefly introduce WINDEX's predecessor, which is called Swiffer. Swiffer is a classification tool for positive selective sweeps. And what's nice about Swiffer is it also can use any input statistics and then using a likelihood framework to learn the joint distributions of these statistics. And so since WINDEX uses the same likelihood framework, and I can also input the same statistics for Swiffer, it was natural that I made these comparisons first. However, the key differences between Swiffer and WINDEX is that Swiffer cannot integrate multiple statistical resolutions, aka the site and the window-based statistics, so I have to do separate runs of that. And it is not a hidden Markov model, so you input all of your data naive of the positions and make classifications from there. So to briefly go over how I ran my experiments, I created realistic human evolutionary simulations with a positive selection scenario and a negative control or a neutral scenario in SLIM version 4.0. I then calculated my choice of site and window-based statistics on these simulation files, trained and tested both tools, and then compared the classifications to understand if WINDEX is in fact a model improvement based off of Swiffer. And so now I'd like to jump into what an example of some of these graphics look like. And again, this is only for one of my simulation scenarios, but I'm happy to talk after the fact about some of the many use cases that I've tested with WINDEX so far. So to orient you all to what some of these graphical distributions look like, the x-axis shows the resolution that I tested with the tool, and then the y-axis shows the proportion of the simulation files that are categorized into each of these classes after I implemented each tool. So for the sweep scenario, I'm going to highlight with this green box. This is the window that contains this true positive location of the selected allele that I put into my simulation data. So WINDEX is performing pretty well at classifying this window correctly as the sweep window in our data. And then it looks like a lot of the windows around this position too are being classified either as linked or sweep, which again, at the site level, usually ends up being mostly linked data, which is what we want to see. If I compare this to a negative control or a neutral scenario, WINDEX does a great job at classifying all of the window data as neutral, which is, again, what we'd like to see with the tool. If I compare this performance to Swiffer, Again, I'm localizing, or I'm just highlighting where the true sweep window is located in this graph. While Swiffer does show, I guess, like the highest prediction bar for sweep being at the right position, it's still a pretty low height for what we'd expect out of that tool. And it also looks like for the rest of the window positions, you're basically flipping a coin and deciding whether or not it's going to be neutral or linked. If you look at the negative control data for Swiffer, there's a low level of misclassification over the entire config. So we can say in general, WINDEX is outperforming Swiffer in this scenario. And then I'd like to dive in a little bit about what's going on at the site level in this data. Graphically, I tried to graph basically the same bars, so it's easy for you all to interpret. But you can kind of think of these as like zooming in on the sites like really close to where the selected allele is in each of these tools. So I can't show all like million bases I simulated, for example. So in my sweep scenario, it looks like WINDEX is overwhelmingly classifying a lot of stuff as linked. We'd expect that sweep bar in the middle of this graph to be higher. But once I show the Swiffer data, maybe there's an advantage, just WINDEX being a little bit conservative here. Once again, in the neutral data, WINDEX is classifying everything as neutral, which is with Swiffer, we see that there's a little bit of a higher proportion of the simulations that are accurately identifying the site position correctly as sweep. However, again, we're facing that coin flip problem where some of the positions around it, even very positionally close, are either being called as neutral or linked. So it's hard to tell, at least from the output of Swiffer, if these sites are truly neutral or linked according to the statistics. And even more concerningly, if we look at the neutral scenario, there's a lot more misclassifications happening both in the linked and the sweep categories. So I'd like to briefly talk about some of the current work that I'm doing with WINDEX. While I'm testing many other simulation scenarios, WINDEX is really designed to be used for real-world positive selection scans. And I've created a set of candidate canonical sites of positive selection from the 1,000 Genomes Project data, which include DARC, LCT, and EDAR, that I'd like to run through WINDEX to understand not only if WINDEX can localize the intended selected allele locations in this data, but also if we can understand the breadth of what these linked regions look like, maybe even get estimates of the proportions of the human genome that are undergoing, or in linked regions are undergoing positive selection. So in summary, WINDEX is a hierarchical HMM that learns from both site and window-based statistics to classify positive selective sweep signatures. WINDEX does well at classifying evolutionary simulation data at the window level, but shows some room for improvement at the site level. And WINDEX is currently being used to scan for canonical sites of positive selection in 1,000 Genomes Project data. With that, I'd like to thank both of my advisors, Dr. Sohini Ramachandran and Dr. Ratamber Singh, my collaborator, Dr. Lauren Sugden, who's also the author of Swiffer, and go check out Swiffer if anybody's interested in the topic. Thank you to Lauren's master's student, Scott McCallum, who laid a lot of the foundation for this project, my undergraduate researcher, Dhruv Raghavan, all the lab members of the Ramachandran and Singh lab, of course, my funding sources. And with that, I'd love to take questions.
Thank you. Yeah, we have some time, like 5 minutes to get some questions.
Can you tell a little bit more about the ratio between the window and the linkage equilibrium around the sweep?
Yeah, I think I'll try to answer what I think you're getting at. It's a little bit of an optimization problem, right? And so what I basically did was I tested a bunch of different window sizes with the simulations that I generated to optimize not only like the signature of the positive selection, but also like minimizing any like muddiness that I see and like maybe using too large or too small.
In your simulation, you put linkage size, right? So and then you pick your window, optimize. How does it change when you change your linkage?
I don't think I've directly explored what you're getting at. Are you talking about like if I'm looking at different simulations, like if
I don't know. The point is that for me, this kind of per site, everything is linked,
Mhm.
and per window, not everything is linked.
Mhm.
It means that there is some kind of ratio between the window size and the linkage of the region that you simulate.
Mhm.
That it's important.
I can kind of understand what you're getting at. I think the way that I've been thinking about it in this model is like the interpretability of looking at windows versus sites. And the usage case of the model is like, if I'm not going to go back, but if I show the visualization where I had maybe some more false positives in WINDEX with the windows, maybe classifying more things as sweep, like I'm willing to sacrifice some of that false positive rate because I think the site classifications are going to be a little bit more important at the end of the day. Then it's more interpretable to be looking at what's going on in the site level than it is the window level downstream. If that helps, like the angle that we were kind of taking with this work.
That's the-
Okay, maybe we can chat a little bit after this.
So I was wondering, when you train your model on real data, how do you get the training data? How do you get the real site that you can train the model on?
Yeah, it's current work of mine, but I can explain like verbally what I'm doing with it. So right now, WINDEX is trained on like a large set of diverse simulations. So the cases I was showing were for like basically like one parameter space of those simulations. But I chose like a demographic model to simulate multiple populations, weak selections, soft like positive selection sweeps, different conditions, so that the distributions that represent going into the model capture as much of these diverse footprints as possible. And so at least for testing the canonical sites, I was going to use the simulation data as the training data and then the real sequences as testing.
But maybe it also depends somehow on the size of the sample or something like that, how many individuals you have.
Yes, definitely. All of my simulations are scaled to the same size as what's in the 1,000 Genomes project. So
Okay.
it should be matching
Okay.
up that way.
Yeah, one more question, maybe.
Well, if there are no other questions, since I have the microphone, I'll ask one,
Okay.
which is I just want to be clear about exactly what you're doing, because you
Mhm.
said you had a 25 site on either
Yeah.
side, and those are variants, correct?
Yes, you're putting variant data into WINDEX. You're not putting like whole genome sequences in.
So is there any information coming in for distance? You're in a low recombination area.
Great
Yeah.
question. Right now, the only, I guess, direct like correction that I'm doing for that is just stratifying by populations that might have different rates of recombination and different mutation rates. But I haven't like directly corrected for that, but it's been like a suggestion I've gotten before to put in like some like distribution to represent differences in populations and whatnot. But also the contigs that I'm simulating aren't huge, and I control a lot of the parameters just to basically evaluate the tool and what I showed you today.
But your polymorphism rates are realistic for the 1,000 genes.
Correct. Yeah.
Okay.
Correct.
I mean, I figured they were, but
Okay, yeah, it's time to move on. And
Thank you.
Thank you for a great talk. Let's